Hello students. Students, today we will discuss a very important topic, and that is translation. We all know that translation is the process of protein synthesis. So what happens during protein synthesis? There is polymerization of amino acid to form polypeptide. So this process of translation is divided into 3 steps, or it is carried out in 3 steps. The 3 steps are, the first one is activation of amino acid, second is charging of tRNA, and the third step is the actual translation process. So first of all, we will discuss the very first step, that is activation of amino acid. We know that there are 20 different types of amino acids which is required for the process of protein synthesis. These amino acids are actually present in the cytoplasm. So what happens during the activation of amino acid? This amino acid will react with ATP and it will result in the formation of amino acyl AMP enzyme complex. And there is release of inorganic phosphate. Just see here, amino acid, it will react with ATP and it will result in the formation of amino acyl AMP enzyme complex with release of inorganic phosphate. This amino acyl enzyme complex, it is actually the activated amino acid. And to carry out this process, we need an enzyme. The enzyme is amino acyl tRNA synthetase. Clear? So you have to remember the name of the enzyme, which the students always forget. The name of the enzyme is amino acyl tRNA synthetase. So the first step in the process of translation is activation of amino acid, where amino acid will react with ATP in presence of the enzyme amino acyl tRNA synthetase, and there will be the formation of activated amino acid, which is actually amino acyl AMP enzyme complex with the release of inorganic phosphate. Now why there is release of inorganic phosphate? We can see here ATP participated and here we have AMP. So that's why there is release of inorganic phosphate. This was the first step. Now the second step is charging of tRNA. This charging of tRNA is also known as loading of tRNA or amino acylation of tRNA. During this process, what happens? This activated amino acid, it will bind to the tRNA, and we have discussed earlier that it binds at the amino acid binding site, which is present at the 3' end. So just see here, amino acyl AMP enzyme complex, which is our activated amino acid. Now what will happen? This activated amino acid, it will bind to the tRNA, and what will happen? There will be the formation of charged tRNA, that is amino acyl tRNA complex. This is actually the charged tRNA or loaded tRNA, and there will be release of AMP and enzyme. If it is not clear, just see this diagram. We have studied earlier that this is the structure of tRNA. Clear? This is the 3' end and this is the 5' end. These are the 3 loops. T loop, this is anticodon loop, and this is the D loop. The 3' end has amino acid binding site. So the activated amino acid, that is amino acyl AMP enzyme complex, it will bind where? It will bind to the 3' end. Clear? So the activated amino acid will bind at the 3' end with its COOH group, that is carboxyl group. So this tRNA will be called as charged tRNA or the loaded tRNA. Is it clear? tRNA. So this was the second step, where the activated amino acid gets binded to the tRNA at the 3' end. Now, the tRNA which is formed, it is known as charged tRNA. The third step is the actual process of translation, which is divided into 3 steps. The first step is initiation of polypeptide chain. The second step is chain elongation. The chain gets elongated. And the third step is when the nonsense codon or the terminator codon comes, what will happen? The process will stop. That is the chain termination. So we will discuss these 3 steps. So just see here. The first step is initiation of polypeptide chain. We know that during this process, what will happen? We need certain things. What are these things? We need the smaller subunit of ribosome, that is 30S. The larger subunit, that is 50S. We need magnesium ion. We need here GTP. And we need messenger RNA. So these are some things which are required during the process of initiation of polypeptide chain, that is 30S, 50S subunits, magnesium ion, GTP and messenger RNA. We know that the 2 subunits, that is the larger and the smaller, they remain free, but during the process of translation they get united, and for that they need the magnesium ion. If the amount of magnesium ion is less, they remain free, and when it is present in more than the needed amount, what will happen? There will be the dimer formation. So here what will happen? These 2 subunits will get united. So this is the smaller subunit and this is the larger subunit. The smaller subunit has mRNA binding site. This is mRNA. So smaller subunit have mRNA binding site, while the larger subunit has 3 sites. That is, just see here, A site, P site and E site. Now what does this A, P and E site stands for? A site stands for amino acyl binding site. P stands for peptidal site and E stands for exit site. Clear? So A site, P site and E site, you have to remember this site in the sequence only. So the smaller subunit, it has mRNA binding site, while the bigger subunit has 3 sites, A, P and E site. Clear? So what happens? This messenger RNA, just see here, the first code will always be the start code. So that is AUG. First code will be always AUG, that is the start codon. Now what will happen? The tRNA will bring the methionine. Clear? It will bring, and it will bring only to the P site. So the first tRNA will always come to the P site, while the other tRNA will come to the A site. It is the exceptional case for the first, that is first tRNA which will bring the message, which will bring the amino acid, the first amino acid, that is formyl methionine. Clear? So the first code is AUG. So the tRNA will come to the P site, that is peptidal site, and will bring the amino acid. Now what happens next? Here we can see there is the next code. It can be any code. Suppose it is GUG. Now what will happen? The specific tRNA will bring that particular amino acid which is coded by GUG. GUG will code for a particular amino acid. So the specific tRNA will bring that amino acid, and it will come to which site? It will come to the A site. Clear? So here we have drawn that the second amino acid will come at the site, or we can say the tRNA will bring the second amino acid at A site. Clear? Now what will happen? There will be the bond formation. There will be bond formation, peptide bond formation between the amino acid 1 and the amino acid 2. The carboxyl group of the amino acid and the amino group of the second amino acid which is present at the site A. There will be the bond formation, peptide bond formation. So here I am writing which two groups. The amino acid which is present at the P site, its carboxyl group, and the amino acid which is present at the A site, its amino group. There will be the bond formation between these amino acids. Clear? Now what happened? Now it's not the messenger RNA which will move. It is the ribosome which will move or which will slide over the messenger RNA. Do remember, it is the ribosome which will slide over the messenger RNA from 5' to 3'. It will slide over in this direction, and this process is known as translocation. This process is known as translocation, and for the translocation an enzyme is needed, and that enzyme is translocase enzyme. Clear? Now what will happen when it will slide over here? We will get a free A site. Now we will get a free A site. Now the next code will be whatever the tRNA will bring, that particular amino acid. Clear? Again there will be the bond formation between the carboxyl group of the second amino acid and the amino group of the third amino acid. And what will happen? The tRNA, now it will get released from the E site. It will exit. Now there is no need of the first tRNA. It will get removed from the site E, and the process will continue. This formation of polypeptide, you can see there is formation, there is elongation of polypeptide. So first step was just initiation, where the tRNA will bring the first amino acid at the P site. Now the second step was chain elongation. During the chain elongation, what will happen? There will be peptide bond formation between the first amino acid and second amino acid. The ribosome will translocate. It will move over it will slide over the messenger RNA, and the process will continue. So there will be chain elongation. And the third step is chain termination. The chain will terminate. The process will stop only when the code will be, the stop codon. When the messenger RNA will bring the stop codon. We have studied that there are 3 stop codon, that is UAA, UGA, UAG. If any of these 3 codon will come, what will happen? The process will stop because they do not code for any amino acid, and the process will get terminated. So that will be called as chain termination. Clear? And we will get a polypeptide chain. So these were the steps of translation. We can see the first step was activation of amino acid. The second step was the charging of tRNA, and the third step was translation, where what happens? The tRNA will bring the amino acid as per the message present in the mRNA. Second, what will happen? There will be the peptide bond formation. This process will continue until and unless we will get a stop codon. So I hope you got this lecture very well, and thank you.